So, dear students, today I'm going to discuss one of the very important topic, that is sequence alignment in bioinformatics. So, till dated, I have talked about what bioinformatics is all about, what are the different databases, the primary databases, the secondary databases, and we have also discussed a lot about the databases. So, as you can take the example of the database, if you consider a database as like a book, right, your library, and in library, a lot of books are there, lot of billions of books are there. But until you don't read them, is there any benefit of keeping the books in the library? Not at all. Likewise, if in the researchers have developed a very good database, and the benefit of the database is, as already discussed, you can share the information throughout the globe anytime, anybody can access it. Anyone can update the data, anyone can submit the data, anyone can retrieve the data. But until biological data is not being analyzed, you will not be able to conclude anything from the biological data. And the ultimate aim of the bioinformatics is to dig out the information, to dig out the knowledge in the biological data which is not explored at, right? So to conclude something from the biological data, we should go for the sequence analysis. So in the sequence analysis, the solve is sequence alignment. So I'm Dr. Nutan Prakash Vishwakarma, and today I'm going to discuss some facts about the sequence alignment. So let's start the sequence alignment. What sequence alignment is all about? So sequence alignment is a way of arranging the sequences of DNA, RNA, or protein. Means in the sequence alignment, we are arranging the sequences of DNA or RNA or protein. Why we are arranging it? So that we can identify the regions of the similarity. Where is the regions where our sequences are showing the similarity? Why this similarity comes? That maybe that comes due to the consequences of functional, structural, or evolutionary relationship between the sequences. Why we are getting the regions of the similarity? Due to their evolutionary relationship. Sequences evolutionary relationship or structural relationship or functional relationship. So another definition, sequence alignment is the procedure of comparing two or more sequences. If you are comparing two sequences, we are calling it as a pairwise sequence alignment. And if you are comparing, you are aligning more than two sequences at a time, that is going to be called as a multiple sequence alignment. Why we are doing so is to search for a series of individual character or pattern that are in the same order in the sequences. So this is the basic two definition of the sequence alignment. What is the importance of the sequence alignment? Number one, functional. DNA molecules that are very much alike or similar in sequence analysis probably have the same regulatory role. If two DNA molecules are sharing the strong level of the similarity, then both may have the same regulatory role. If you talk about the protein molecule, if two protein molecules are very much alike, so probably they have same biochemical function. So suppose you have a novel DNA molecule, if you have a novel DNA sequence, and you want to know the structure of, function of that particular DNA. So rather than going in the wet lab, what we do, if we align our DNA sequence from the existing database, and fortunately, if your sequence is matching very strongly with the sequence already available in the database, you can assign the same function to your novel DNA sequence. Right? Likewise, if you have a novel protein which you have isolated in your laboratories and go through the sequencing, once you get your sequences, you match your sequence, you align your sequence with the existing databases, protein sequence databases, and if your sequence is showing very strong level of similarity, then even without moving in the wet lab, you can assign the same function to your novel protein. So you can save a lot of time and effort. So importance number two, the structure. Protein molecules that are very much alike probably have the same 3D structure. As in previous lectures, I have discussed a lot about the protein 3D structure, that to in the structural bioinformatics or in the structural biology, we study about the 3D structure of the proteins. Why it is important? 3D structure of protein is very much important in the structure-based drug discovery or to understand the mechanism of the enzymes, right? So, how you can determine the structure of any particular protein? There is three methods available. Number one, X-ray crystallography, NMR, and electron microscopy. So most of, if you go on the PDB, I think you already studied about the protein data bank. More than 95% of the structure is solved through the X-ray crystallography. Very few from the electron microscopy, NMR, and very, very less by the electron microscopy. So solving the structure by the help of the X-ray crystallography and NMR and electron microscopy, it's a too much time, effort and time consuming, and it's a difficult task. Suppose you have a current problem, you want to solve it. Scientists have the problems like just current COVID-19 situation. If scientists want to make a particular drug which is effective against coronavirus, if they go for the protein structure solving in the wet lab, it is going to take a lot of time. So if they want to solve this particular structure in the quick way, they can go for the protein structure modeling. In the protein structure modeling, if your novel protein of which you want to determine the 3D structure of it, you want to solve the 3D structure of it, what you can match your this particular protein sequence, your novel protein sequence, with the database of the protein sequences whose structure is already solved and being submitted in the PDB. So if your novel protein is showing strong level of similarity, you can assign the same 3D structure as your template structure have in the protein data bank. All right. So that's how protein molecules that are very much alike probably have the same 3D structure as your template has. The importance number three, the evolutionary relationship. So if two sequences from different organisms are similar, then there may have been a common ancestor sequence, and then the sequences are then defined as being homologous. All right. So if two sequences have the common ancestor, then obviously both the sequences should show the strong level of the similarity, and in that case, the sequence is going to called as a homologous sequences. So the alignment indicates the changes that could have occurred between the two homologous sequences and a common ancestor sequence during evolution. If you do the uh, alignment between two sequences, so definitely you can track that during the course of the evolution, where is the changes, where is the mutations, maybe it is substitution or matches or mismatches, right? The insertion or deletion, so that you can track. So what are the different terminologies which we use frequently during the sequence alignment? So number one is the sequence identity. So once we talk about the sequence identity, it is the matter of protein or DNA. Both have a different meaning. In the sequence, once we use the sequence, that two DNA sequences or two gene sequences are showing 80% of the sequence identity or 80% of the sequence similarity. Both have same meaning. While we are dealing with the protein and DNA, there is no difference between the sequence identity and similarity, right? So either I can say that two gene sequences are 80% similar, or I can say two gene sequences are 80% identical. Both have the same meaning. But once it is the matter of the protein, both have the different meaning. While we are talking about the protein sequence alignment, If I say that two protein sequences are 80% identical, means both the protein sequences should have the exact amino acid as sequence number 1 have. The sequence number 2 also should have the same amino acid in 80% of the cases, 80% of the positions. But while we are talking about the sequence similarity means not exact. Obviously, we are going to include the identical sequences. Along with that, we are also taking the similar chemical properties amino acids. So, for example, if you take the example, suppose for the with the sequence number 1, at the position number 1 you have the glycine, right? And the sequence number 2, at the position number 1, glycine is also there. So this is simply known as identical. At the position number 2, sequence number 1 have the glycine. At the position number 2, sequence number 2 have the alanine. So obviously you can call it as the mismatch. But in the case of the protein sequence, you can call it as a similar, because if you see the structure and chemical properties of glycine and alanine, so glycine and alanine both have the common chemical properties. So in that case, you can say that glycine and alanine are the similar sequences. All right. So once you are talking about the sequence identity, both sequences should have the exact amino acid. But while we are talking about the sequence similarity, identical sequences we are considering, plus similar amino acid we are also considering. So in that case, you can conclude that sequence similarity may be more than sequence identity. All right, either will be equal to identity, or it will be more than the identity. And as already talked about the pairwise alignment and multiple sequence alignment, once you have two sequences at a time and you want to align them, that is simply going to be called as a pairwise sequence alignment. And if you have at a time more than two sequences with you and you want to align, that is going to be called as a multiple sequence alignment. Right, very simple. Two sequences at a time, that is known as pairwise. More than two sequences at a time, that is going to be called as a multiple sequence alignment. All right. So sequence similarity we have already talked about. So on the other hand, similarity is a direct result of observation from the sequence alignment. Sequence similarity can be quantified using percentage. Obviously, we can quantify it, that whether if your two sequences are similar, then how much percentage of the similarity both sequences are sharing. So homology is a kind of the qualitative statement that whether two sequences are homologous or not, means both the sequences are common ancestor or not. But sequence similarity, sequence homology is the qualitative statement. Only we can say that two sequences are homologous or not. But once you talk about the sequence similarity or identity, you can quantify it, that if two sequences are showing similarity or identity, then how much percent? How much is the percentage? So, for example, one may say that two sequences share 40% of the similarity. Right? Two protein sequences are 40% similar. So it is incorrect to say that two sequences are share 40% homology. We can't say that if suppose two sequences are sharing 40% similarity, so they are homologous sequences. No, we can't say. There is certain rule. If you follow those certain rule, with the help of that rule, you can say that doesn't matter how much percent similarity two protein sequences are sharing, whether they are homologous or non-homologous, right? So let's see those rules on which ground we can say that two protein sequences are homologous or not homologous. See this particular graph. See this particular diagram. Here on the x-axis I have put a sequence length, and on the y-axis here the sequence identity, right? So if your two protein sequences, you just try to understand, if your two protein sequences are having 150 amino acid length and both are sharing 40% identity, all right, how much percent? 40%. Both the protein sequences have 150 amino acid, and once you align them, you come to know that both the protein sequences are sharing 40% identity. So if I just go upside from the x-axis and here from the y-axis, and where both the points intersect, here you can say you can put a particular dot, and that dot here you can see that in the safe zone there is the 3 zone. So my dot for the 150 amino acid length and 40% identity is comes somewhere here, right? So that dot comes under the safe zone. So if dot comes under the safe zone, you can say that both the protein sequences are homologous. So now you can conclude that if two protein sequences having 150 amino acid length, and if you align them, you come to know that both having 40% sequence similarity or identity. Here I'm emphasizing the sequence identity. Then you can say they are being put in the safe zone. So you can say they are in the homologous sequences. Now let me take one more example. If I ask you that if two protein sequences both have the 50 amino acid length and both the protein sequences are sharing 40% identity, can you say that these two protein sequences are homologous? Let me help you to understand. Suppose 50 amino acid length, all right, and 40% sequence identity. So if I go and align, you come to know that somewhere this is the point where your dot comes. So in that case, you can say that your dot comes under the twilight zone. So if your dot comes under the twilight zone, you are less certain about their sequence homology. You can't say they are homologous, and you cannot say they are homologous or not homologous, right? You have to go for further validation. So you can't say in this case. Let me give you one more example. If I say that two protein sequences having 50 amino acid length of each, and if you go for the alignment, you came to know that two protein sequences are sharing 35% of the identity. Can you say that two protein sequences are homologous or not? Let me help you. So your dot will come here. So that is again the twilight zone. So again you can't say they are homologous. And in the midnight zone, this is known as midnight zone. If your dot comes under the midnight zone, it clearly indicates that it is damn sure that two protein sequences are not homologous at all, right? So whenever you have to say that two protein sequences are homologous or not, you have to follow this particular diagram. All right. So I think it is clear to you. So sequence analysis, sequence homology. So when two sequences are descended from a common evolutionary origin, they are said to have a homologous relationship or say homology. So sequence homology is an inference or conclusion about a common ancestral relationship drawn from sequence similarity comparison when the two sequences share a high degree of similarity, as we have already discussed. So, lot of terminologies comes in the lot of questions comes in the competitive examination. Maybe it is PhD entrance examination, or you are going to appear for the CSR net examination, or the PhD related examinations, or the biotechnology eligibility test. You can find certain questions related with the homology. So let's discuss few of the concepts. So homologous genes are genes that derive from a common system. Very simple. Homologous genes can arise through a variety of different biological processes. So very simple, homologous genes are those genes which derive from a common ancestor. So what are those events? What are those, due to which your genes are derived from a common ancestor? What are the reasons? So two things may be there, two biological processes may be there, due to which your protein sequence, your gene sequence shows the homology, and they separate from the common ancestor. So those two are, number one is speciation and number two gene duplication. So pairs of homologous genes derived by speciation which result in two homologous genes diverging in different lineages are described as orthologous and called orthologs. Means if your two genes are separated and they become the different genes due to speciation, that is known as orthologous genes. And if homologous genes arise by gene duplication. If gene duplication takes place, and they are going to be called as paralogs or paralogous or paralogs. Right? So if homologous genes derived by event speciation, we call as orthologous genes, and if genes arise by event gene duplication, in that case they are going to be called as paralogs, and orthologs plus paralogs both are the example of homologs, right? So homologous genes of two types, orthologs and paralogs. As you can see very clearly, speciation. Speciation means genes are in the different species. Say for example, like our hemoglobin gene of the human and hemoglobin gene of monkey, right? So we and monkey both have the common ancestor somewhere, sometime lakhs of years before. Speciation happened, and monkey have the hemoglobin gene, and we also have the hemoglobin gene. It's due to speciation, our hemoglobin gene and monkey's hemoglobin gene are different. One more example I would like to give you. Suppose we talk about the genes in human being, like BRCA1 and BRCA2. So BRCA1 and BRCA2 both are responsible for the breast cancer in the woman. So two copies of the BRCA is present, right? So how two copy comes? Due to the gene duplication. So simply you can say BRCA1 and BRCA2 both are paralogs, and definitely their ancestor was same. So you will find the strong level of similarity or identity, right? So orthology term was given in 1970 by the molecular evolutionist Walter Fitch. Sequences which are inferred to be descended from the same ancestral sequence, separated by a speciation event when a species diverges into two separate species. What is the speciation? If you want to go in detail, speciation is the phenomenon during which a common ancestor gives birth to two subgroups that slowly drift away from their common genetic makeup to become distinct species. So biologists usually expect ortholog to have similar function and structure, right? So all the orthologs have the similar function and similar structure. If you go for the observation of the human globin gene and monkey's globin gene, you will find very strong level of identity, and both have the similar function and structure. So let's take one more example. Here you can see, suppose one gene was there in the common ancestor, the event happened, duplication. Due to duplication, gene A and B, right? And speciation takes place. So A1 and B1, A2 and B2. So whenever there is the speciation, that is known as orthologous. And wherever there is the duplication, there is the paralogous. So here you can see that A and B, right? A and B both are what? Paralogs, right? Just like BRCA1 and BRCA2. And if you see the A1 and A2, so how is A1 and A2 formed? Due to speciation. So here in this case, A1 and A2 both are going to be called as a paralogs. Here, let's take the example of one example, like Arabidopsis thaliana and Chlamydomonas. Arabidopsis thaliana, it comes under the higher plant, and we use as a model organism, right, in a lot of genetics related research. And it's a Chlamydomonas, a single cell algae, right? So one protein is there that is known as flu regulatory protein. So flu regulatory protein is present in both in the Arabidopsis thaliana and in the Chlamydomonas. But if you see the structure and sequence, and if you analyze the flu regulatory protein of Chlamydomonas, it is very complex and it crossed the membrane twice. However, the Arabidopsis thaliana's protein cross membrane only once. Along with that, the Chlamydomonas proteins have some additional domains, and it undergoes alternative splicing as well. However, if during the genetic engineering experiment, what scientists have done, they replace the Arabidopsis thaliana fluororegulatory protein, fluororegulatory gene with they replace it with the fluororegulatory proteins related gene in the Arabidopsis theriaca. Although there was the huge difference, lot of difference in the structure, but still the proteins of Chlamydomonas completely substituted the function of Chlamydomonas. Means it works fine. So if transferred from algae to plant genome by means of genetic engineering, significance means, however, it can fully substitute the much simpler Arabidopsis protein. If transferred from algae to plant genome by means of genetic engineering, it means it can clearly, it is mentioned. So significant sequentiality and shared functional domain indicate that these two genes are orthologous genes inherited from the shared ancestor. Alright. So paralogy or paradox. As we have already discussed, the paradox genes arise due to the event of duplication. I have already given the example. Like here in this case, the A1 and B1, right? So A1 and B1 are paralogs because in the same gene, no, in the same organism, two genes are there, A1 and B1. And since there is a duplication, so wherever there is the gene duplication, their function may be different because their locus change, right? So paralogs are homologs separated by a duplication event, meaning that within a genome, a gene was duplicated. One of the duplication may have kept the original function, while the other duplicate could have acquired a new function. But in the orthologous genes, the structure and function was conjugate. But in the paralogs, their original the original one will keep its original function, while the other one could have acquired a new function. So mechanism of gene duplication allows these sequences to have evolved different functions. Okay. So for example, the best example of the paralogs genes are the four known classes of hemoglobin: hemoglobin A, A2, hemoglobin B, and hemoglobin F are paralogs of each other. While each of these proteins serve the same basic function of oxygen transport, right? Maybe it is hemoglobin A or A2 or B or F. Both all have the same basic function, that is carrying the oxygen. They have already diverged slightly in function, but due to gene duplication, they have diverged in function. Like fetal hemoglobin, that is called as the hemoglobin F, has a higher affinity for oxygen than adult hemoglobin. So as I already told you, that if paralogy takes place, the function may slightly change. Alright. So here you can see one more example, like homologous and orthologous. Homologous, under the homologous, the two things are there, orthologous and paralogous. If suppose one early globin gene was there, gene duplication takes place. So alpha chain gene and beta chain gene, right? So these two are known as what? Paralogous, because gene duplication takes place. So frog alpha and here you can say the mouse alpha. So how frog and mouse originated? Due to the speciation. So frog alpha and mouse alpha is known as orthologous. Frog alpha, chicken alpha, mouse alpha are orthologous. Mouse beta, chick beta, frog beta are known as orthologous. But if you talk about the mouse alpha and mouse beta, right, they are two copies of the alpha and beta in the mouse. They are going to be called as a paralogs. So I think it is the concept of the orthology and paralogy is very clear to you. One more example, the analogy. What is analogy? Analogy, if similarity between sequences arise due to convergent evolution, those genes are called analogous, right? Their functions is same. Somehow during the course of evolution, now their function is same. But if you see their sequences, it is totally different. But function is same, right? So I would like to quote some example, just like the bat of wings and wings of bat and wings of birds. Their structure is same, but their function is same. Both bat and birds use the wings for flying, right? So there is the convergent evolution. Although there is a huge difference in the structure of uh, wings of bat and birds, but the function is common. Likewise, in the genes, if they are not showing the strong level of similarity, but if still their function is somehow conserved, that is going to be known as convergent evolution. All right. So if similarity between sequences arise due to convergent evolution, those genes are called analogs. So convergent evolution create analogous sequence that have similar form or function but were not present in the last common ancestor of these groups. In 2013, the first genome-wide study of convergence was published. Comparison of the genome of echolocating bat and the dolphin identified numerous convergent amino acid substitution genes and implicated in hearing and vision. Means the genes related with the hearing and vision, although they were not showing a strong level of similarity, but still the function is same. So analogy comes due to the convergent evolution in which gene sequences are not sharing strong level of similarity, but still their function is same. One more example like xenology. What xenology is all about? So homologs resulting from horizontal gene transfer between two organisms are termed as xenolog. Two types of the gene transfer is there. One is known as vertical gene transfer, another is known as horizontal gene transfer. What happened in the case of the vertical gene transfer? Like we people, like my father, my forefather, right? So genes comes from my father to me. In my father's genes came from the my grandparents, right? So likewise we have the one hierarchy: grandfather to father to son, then my son, then their son, right? So likewise that is simply known as vertical gene transfer. What horizontal gene transfer is all about? In the horizontal gene transfer, I think you have heard about the transformation, transduction, and conjugation in the bacterial, in the monomer, right? So if transformation takes place, one bacteria taken the net DNA from its environment. No one can say from where that particular DNA came inside the bacteria. So such kind of transfer, horizontal gene transfer is known as horizontal gene transfer. So gene is a Greek word that means foreigner. That is also coined by Walter Fitch. So genolog result from a lateral transfer between two organisms, a direct dynamic transfer between two species. Xenologs can have different function if the new environment is vastly different for the horizontal moving gene. Means whatever gene you are transferring from one organism to another one. So definitely if organism change, then atmosphere will change and locus will change. So that may gain a new function. Let me take a example. A typical transfer of lateral transfer or genolog is the acquisition of the isoleucyl tRNA synthesis. This is you on isoleucyl tRNA synthesis one protein from their host by several bacteria. The isoleucyl tRNA synthase is a protein involved in the synthesis of other protein, obviously. And it is acquisition by bacteria. If any bacteria acquisite from its source, means host, so from host, it accumulate inside its DNA. And its acquisition by bacteria seem to help. Now, previously the isoleucyl tRNA synthase was helping in the synthesis of proteins, but once it is acquisited by bacteria, now its function is going to be changed. What function it carries? Now it start working as to gain the antibiotic resistance in the bacteria. When this happens, the newly acquired isoleucyl tRNA synthase is a xenolog of other tRNA synthase contained in the bacteria. So this is simply known as genology. So I think this is the enough concept for today. In the next lecture, we will go in detail about the sequence alignment and we'll learn about certain algorithms that how to solve the problems of sequence alignment. So if you like the video, you can hit the like, you can share with your friends so they can also understand the concept. And obviously you have to, if you are new in the video, you can subscribe it so that whenever I upload any kind of video, such type of video, you will get the notification. So thank you all. Take care. See you in next video. Jai Hind.